Welcome to Integrating Pathology and Next-Gen Sequencing for a Robust and Practical Workflow. I'm Tamlin Oliver, Managing Editor of Genetic Engineering and Biotechnology News, and I'll be the moderator for this webinar. Our speakers today, two of them leading pathologists, will talk about why you should build a clinical oncology next-gen sequencing program, give tips on how to do it effectively, and then address the challenges specific to establishing a robust oncology NGS workflow. If time allows, they will also be available to answer your questions on best practices. Before the webinar gets underway, though, I want to introduce everyone. Our first speaker will be Mike Feldman, Associate Professor of Pathology and Laboratory Medicine, Director, Cancer Center Core, Tissue Bank Hospital of the University of Pennsylvania. Bob Daber, Technical Director of Clinical Genomics, University of Pennsylvania School of Medicine, will speak second, and they will be followed by Clifford Hoyt, Oncology Fellow, Tissue Applications and Collaborations, and Andrew Berry, Senior Manager of Biomolecular Applications at PerkinElmer. Mike, we have a lot of material to cover today, so I'm going to have you get started right away. The floor is yours.
Thank you very much, Tamlin. This morning, I'd like to talk to you about the role of anatomic pathology in the next-gen sequencing of solid tumors. So why build an oncology program at all? Well, we know that cancer is a disease of genetics. It's caused by a variety of mutations, insertions, deletions, amplifications, rearrangements, and large-scale aneuploidy of the genome. It occurs across multiple genes, and all of these have implications for the prognosis, diagnosis, and eventual therapy selection that we can offer to treating clinicians. But why go with the next-gen sequencing panel rather than a single-gene assay? Well, it really comes down to efficiency and the ability to look across a large variety of markers simultaneously. The single-gene assays are where we started years ago, and what we're seeing is that the complexity of the human genome and the changes in that genome in oncology are really not able to be kept up with one gene at a time. And so we're really looking at moving to panels of genes and eventually to exomes and genomes in the distant future. But the ability to analyze many genes simultaneously to look for nucleotide variations, deletions, copy number, and rearrangements allows us to push work through faster through the clinical labs, as well as have a much higher sensitivity than the original single-gene assays, which relied on Sanger sequencing. That said, it's not simple to do these types of assays. They're very complex. The analysis of these is complex. The genomic changes within the tumor cells are complex. The samples are very heterogeneous. A tissue sample is not pure. It's not a pure tumor. It contains inflammatory cells, stromal cells, endothelial cells, areas of necrosis. So all of these make the analysis by next-gen sequencing complex because of the heterogeneity inherent within the tissue. And formalin fixation, frankly, adds a layer of complexity because of the degradation of DNA that occurs. And finally, how we report all of this to get this information back to clinicians is actually quite complex and is still an area where we are evolving that process. In the end, what we're looking to do is to provide clinicians who are treating patients with the information and knowledge they need to make the best choices for targeted therapeutics. So where should anatomic pathology be involved in this entire process? At Penn, we place anatomic pathology at the very beginning of the analytic pipeline to allow and facilitate the best tissue selection for the downstream analysis in the next-gen sequencing lab. It's the old adage that garbage in, garbage out. If you don't have a high-quality sample going into the analysis that has the most tumor with the least amount of necrosis and the least amount of interfering non-tumoral cells, it's going to hamper the assay and its ability to pick up small variations in the genetic material. It's also not uncommon for patients with tumors to have multiple samples, so finding that best sample may require you to not only look in your own hospital, but it may require you to go to outside institutions to find that best sample to test. Once you've identified the sample to test, the anatomic pathologist needs to analyze the abundance of tumor within that sample. Next-gen sequencing relies on the ability to call variants based on the amount of tumor that's actually present within a sample. And very, very small samples of tumor that have low abundance of tumor relative to stroma can actually make the next-gen sequencing ability to call variants that much more challenging. And then finally, we need to be able to tell the next-gen sequencing lab within the sample how much necrosis is present, because necrotic tissue is not going to have amplifiable DNA. The quality simply will be too poor. So for us, the process starts with allowing clinicians to place electronic orders. We use the Epic order system, and that order comes to the anatomic pathology lab, where we go into a case selection mode to identify the best material. And then based on how much tumor is present in that material, we either create paraffin rolls or slides that can then be macro dissected to scrape into an Eppendorf tube for DNA extraction. Once that occurs, a message goes back to the electronic order system that the sample's been identified and moves downstream to the Center for Personalized Diagnostics for nucleic acid extraction and next-gen sequencing, which Bob is going to speak at great length. In anatomic pathology, we still have to determine how much tumor is present within a DNA sample. And what you can see in this graph is that there's a tremendous amount of inter-observer non-reliability, if you will, of identifying how much tumor is present. You can see across a variety of tumor samples, different pathologists telling you with absolute certainty how much tumor is present in each of these samples, and the range is quite staggering. So we started to ask if there was another way of monitoring and analyzing for the abundance of tumor in a pathologic sample. We began this using an automated machine analysis algorithm developed by PerkinElmer called Inform. And what I'd like to show you in the next slides is examples of how we used Inform and compared Inform to the visual estimates of anatomic pathologists, as well as the allele frequency estimates from the next-gen sequencing. So the first case I'm showing you is a melanoma. It's a metastatic melanoma, where the anatomic pathologist estimate was greater than 50% tumor. And what you can see circled in red is the area that was macro dissected, and by next-gen sequencing was only about a 30% tumor estimate. So the visual estimate seems to be a little bit higher than what was actually present. This is the second case, again, where the visual estimation of tumor was about twice what the next-gen sequencing analysis demonstrated. And just a little higher power to show you the complexity of these fields of view, that these are not pure tumor, that there's mixtures of inflammatory cells, and visually estimating how much tumor and how much inflammatory cell is present is actually a very challenging exercise. Across a range of tumors, this table shows you in red areas where there was very good correlation between the pathologist estimate and the next-gen sequencing allele frequency estimates. But in black, we're showing you where there was significant discordance, 2 to 4-fold. And it was this discordance that really pushed us to ask, would an automated machine algorithm help or streamline this process? The tool we chose to use is a machine learning algorithm that Cliff is going to spend more time talking about from PerkinElmer. This is a tool called Inform, and it relies on the ability of machine learning algorithms to identify tumors within a section compared to the stroma, and then find all the nuclei within those two compartments, and then provide an estimate of how much tumor nuclei are present relative to the stromal nuclei to give you percent tumor. And this is an example of where the Inform analysis finds the tumor highlighted in this brown, and then allows you to count nuclei present within these regions relative to the stroma. The first thing we did in this study was to just take a series of cases where we had allele frequencies across a range of different tumors and compared Inform to a manual gold standard method where we took field of view and counted the cells one by one, painstaking process that one of my residents and I took. And in that analysis, we saw that the ability of Inform to measure the abundance of tumor correlated very nicely with manual counting, with a linear regression of about 0.96. Across those 9 cases, the Inform analysis shown in the far right was compared to the allele frequencies identified in the next-gen sequencing panel. That's the column called CPD, compared to single-gene analysis of allele frequency, and then finally the anatomic pathologist visual estimate for each of these cases. And what you can see very nicely is that the Inform analysis correlates quite nicely with the allele frequency that was identified either by pyrosequencing or next-gen sequencing. And in several cases, actually correlated better than the visual observation by the anatomic pathologist. These are examples of what Inform does. So in this first case, we have a colon carcinoma. Manual counting was 42% tumor. The machine counting by Inform was 39%, and the sequencing analysis was 50 to 60% by either pyro or CPD. And the anatomic pathologist estimate was about 26 to 50%. They all correlated pretty well. The next case is a metastatic melanoma, where the anatomic pathologist estimate was quite low, in the 10 to 50% range. Allele frequencies for these tumors were near 100% by either single-gene sequencing or next-gen, and both the Inform and manual showed that these were nearly pure tumors. So a significant variation between the manual count by the pathologist and the automated counts by the machine learning algorithm. And here was another one where the anatomic pathologist underestimated the tumor abundance, where they were scoring it at around 10 to 25%, and the allele frequencies by gene sequencing were in the 40, and by machine learning were as high as 70%. So we see the opportunity to use machine learning as a promising tool, but it still suffers from some workflow issues. It does a very nice job in terms of quantifying tumor percentage, comparable to a gold standard manual counts. There's still some issues with the ability to segment tumor nuclei and stromal nuclei, but that's being iteratively improved with feedback. And we see this as an opportunity to use this as a tool within the workflow of the anatomic pathologist. So rather than relying on just a quick visual inspection, actually allowing a digital slide of the material that's going into the next-gen sequencing to be automatically quantitated using Inform, not as a replacement for the pathologist, but actually as a tool that they can have in their workbox. And with that, I'm going to turn it over to Bob to take you through the next-gen sequencing pipeline.
Bob, before you get started, though, I do just want to remind everyone about our Q&A session following all the presentations, and please feel free to submit questions at any time through the Ask a Question box. All right, Bob, take it away.
All right. Thank you so much for the solid introduction about the importance of the samples upstream of next-generation sequencing. What I'm going to do is spend a little bit of time talking about the workflow as it relates to DNA extraction through the final report. And these are topics that in itself could be hours upon end. So I'm going to give a high-level overview and really walk through our experience in building the laboratory and then the hurdles that were revealed in that process. So if we take a look at a clinical next-gen assay, there are a couple components that I'm not going to chat about, the first being tissue review, which Mike has just described, and then on the back end is reporting these results into the electronic medical record. What I'm going to do over the next few minutes is talk to you about the steps in between as they relate to the molecular lab, essentially DNA extraction, the library preparation, the sequencing, and the bioinformatic analysis. And so the DNA extraction is critical. A lot of these specimens, we end up with scant tissue, so small amounts of tissue. And the challenge is to try and identify the methods that allow for the highest yield of DNA with the greatest purity, since these assays are so sensitive in terms of mutation allele frequencies. We're sequencing single DNA molecules, and so any background errors associated with that or the fixation process really define the lower limits of detection. After extraction, we enter library preparation, and library preparation is essentially asking the question, if we start with the entire genome, right, all of the genes of the genome, which areas of the genome do we want to sequence? And we can either do whole genome sequencing, we can do exome sequencing, we can do targeted panels. And so there are a variety of different methods which allow for the capture of those regions of interest. On the sequencing side, this is now taking those fragments that have been enriched and turning them into digital data, the process of sequencing. And this is really what revolutionized this field. These new sequencers, these machines are able to sequence millions to billions of individual DNA molecules, resulting in a very large amount of data, which is where the bioinformatic analysis comes in. And this is the process of taking essentially images or signals of different nucleotides for each of those strands, placing them back into the context of the genome and identifying where the variation is that ends up in the ultimate report. And so this is quite challenging, and many labs are wrestling with that component. And I'll talk to you about some of the things we've done to try and overcome those barriers. On top of all of these different components, there's a lot of commercial competition, so there's a lot of different options. And as you're starting to build a laboratory, as we did about a couple of years ago, it was very difficult to try and identify which of these technologies was the most robust. And about every 6 months, we're seeing a turnover. And so in the very beginning, we tried to get our hands on as many different forms of this technology as possible to identify which one would provide us with the most robust workflow, really fitting in with that mission of trying to identify as many mutations as possible so that our clinicians could really make informed decisions. On top of all of the different commercial options and those choices, there are a lot of operational variables that go into place, all surrounding the clinical test. And so to our clinicians and our providers, they see a test. Inside the laboratory, we see all of these different steps, and there are many different variables that need to be balanced to generate an assay that meets their needs in terms of turnaround time, in terms of cost. And so here are just some of the questions that we need to address as we look to building one of these technologies. The first, and probably one of the most challenging, is what is insurance going to pay for? Is this test going to serve the clinical community as well as the research community on campus? And therefore, we might need additional genes present on this panel to fuel the research endeavors that are taking place. Do we develop universal panels, or do we develop tumor-specific panels? And this really depends upon questions such as what's the total volume for each of these tumor types? How often do we need to update content? How do we select that content? Balancing things like reagent shelf life. And then really, from a turnaround time perspective, in the clinical setting, these results need to be out in less than 2 weeks. So that means we need to have the appropriate batch sizes. We can batch every single day and get results out pretty quickly, but that's going to cost more money. Essentially, it's like looking at a dishwasher. You can run the dishwasher when it's not full, or you can wait until it's full. And so operationally, those are challenges that we need to figure out. On top of that, because we're doing this in a clinical setting, the assay needs to be so robust that we're able to achieve reproducible results from run to run. And so that requires us to have an automated or very high reproducible workflow that allows us to scale as demand scales and to create this clinical grade data. On top of that, there are a lot of moving pieces and a lot of different pieces of data that all need to be integrated in order to really understand the final results. And so across this workflow, we need both automation as well as laboratory information management systems to be able to track all of that data. All of these need to fit into the requirements set by CAP. And so these are new requirements that came out as this technology was emerging into the clinic. As you can see, there's a large number of them touching on both the wet lab components and the bioinformatics. And all of these need to be kept in mind as you're developing a given assay if you're working underneath in a lab that uses CAP for its accreditation. So how do you get started? Putting all of these pieces together is just like a big puzzle. You know what it's supposed to look like in the end, right? You've got the cover of the box, and you've got all these pieces. And getting started, is challenging until you just start putting the pieces together. And so one word of advice that I have for most labs looking to get into this space is just get started. Start getting experience with the different components of the workflow, and from there, you'll gain the knowledge and experience you need to build the assay. So what did we do in terms of our development roadmap? Really broke it down into 4 different phases. The first is the piloting phase, and that's where we got to test out all the different technologies to try and understand which ones met our clinical demands and needs. After that, we locked down our assay and entered into what I call the optimization phase. This is where we go back to the protocols, go back to our workflow, and start asking the nitty-gritty questions about the relationship between all of the different moving pieces to try and come up with protocols that are reproducible and that really push our limits of detection. After that, we move into the validation phase, which is taking those SOPs and taking those protocols that we've developed and just asking the question in a blinded fashion, how well does our test work in terms of assay sensitivity and specificity? And after that, we can transition it into a stable test. So for us, after doing the piloting phase, we made a few decisions about our workflow. On the DNA extraction and QC side, we tested a variety of different extraction kits, and in the end, we decided to go with a homebrew-based method, which was a mix of a QIAGEN kit with pure gene, followed by QC performed on instruments from Agilent, and then a qPCR assay. On the library prep side, we went ahead with an amplicon-based approach, and this was the TruSeq amplicon assay from Illumina, with subsequent sequencing on the Illumina MiSeq. And in the end, for the bioinformatic analysis, we decided to go with a homebrew assay using tools that we developed in-house, as well as several academic and commercially available options. Why did we choose to go that way? Why did we choose these different components? Well, on the extraction side, it's really a challenge. As Mike had said, it's garbage in and garbage out. And that holds true with DNA extraction as well. If we can't get pure DNA in reproducible yields, then it doesn't matter how robust our assay is downstream of that. We really need to be able to produce high-quality data, and we need to have tight control over the final concentration. For these really small tissue specimens where the DNA is limiting, being able to control the rehydration volume is important so that the concentration of DNA is sufficient for the library prep. In terms of library prep, we chose to go the amplicon-based approach because it worked well with FFPE DNA, the DNA that was fixed in formalin. It allowed us to have low input amounts, so we can go down to about 150 to 250 based upon the manufacturer's spec. And I'll show you in a little bit how we're able to push that even lower once we entered our optimization phase. In terms of turnaround time, these assays could be performed within a single day. So coming in at about 8 o'clock, we can have libraries on the sequencer before the end of the working day, which is great, allowing us to achieve about a 3.5 to 4 day turnaround time in the best case scenario for our specimen. And the assay was also scalable, allowing us to do up to 96 samples in a day with very high capture reproducibility, meaning we're able to determine copy number from this assay. On the sequencing side, we chose to go with the MiSeq because of the ease of use, only about 30 minutes of hands-on time, high-quality data, and we do perform paired-end reads of 185 base pairs by 185 base pairs, and this allows us to take each of our reads and have full overlap in both directions, therefore removing any type of sequencing artifact in the downstream informatics. On the bioinformatics side, we chose to go homebrew to give us full control. This allowed us to get higher sensitivity by developing different software tools, different algorithms that really push the envelope and push the limits of detection. And then we were able to integrate this into what we've built in terms of a homegrown lens that allowed us to do everything from sample tracking all the way through the final report generation. So this was our menu. After just looking at the different panels that were available, we decided to build a custom heme malignancy panel composed of 33 genes, specifically looking at AML, where genes such as ASXL1 and TET2 are important in the prognosis. And as you can see, we're not present on the solid tumor panel that we were using. And this solid tumor panel is an off-the-shelf solution developed by Illumina with nearly identical content to the AmpliSeq cancer panel. So after ending the piloting phase and locking down that assay, we moved into optimization, where we asked the question, how do we tweak these assays to achieve the maximum performance? Again, looking back at the 4 different components of the next-gen assay as it related to our workflow inside the laboratory, DNA extraction, we knew we can get pretty good yields, but we wanted to push it further. The same thing with library prep. Out of the box, it worked well, but we wanted to define better the relationship between DNA quality and the actual preparation steps. Sequencing did not require much effort, and bioinformatic analysis was a pretty uphill battle. So jumping now into DNA extraction and the relevant QC. During our piloting phase, we noticed that our formalin-6 DNA samples did not all have the same assay performance, meaning we'd put in the required amount of DNA that the protocol specified, and in this case, either 150 nanograms of high-quality DNA or DNA from fresh tissue, or 250 nanograms from formalin-6 tissue. And over on the right here, you can see what ends up happening after we performed library prep and generated the data. There was a fraction of samples where we did not have enough DNA present after extraction, so we couldn't even run the assay. And then we had 4% of samples that failed the assay no matter how many times we repeated it. The remainder of the samples either gave us high-quality data, which was comparable to using DNA from fresh tissue, or low-quality data. And what I'm using as the definition here is essentially the background error rate. So with our high-quality data, we saw an error rate of maybe 1 to 2% in terms of allele frequency. So by setting a threshold of 5% in our variant calling, we had no false positives arising from those samples. Whereas on the low-quality side, we saw variants somewhere in the 8 to 15% range. And if we'd repeat analysis, those variants would disappear and new ones would pop up. So we knew that those were false positives, somehow associated with something in the assay or with the DNA itself. So one of the first things we did is said, well, maybe just measuring the concentration of DNA on a fluorescent-based measurement is not enough. And so we looked at running a gel electrophoresis. And so this is a human DNA gel, goes about 50 kb down to 200 base pairs. And you can see right in the middle is blood DNA. You get a nice tight band right above 50 kb. The rest of the bands on this gel, you can see smears. And these all correspond to different FFPE samples. The ones over to the right being the samples that continually failed prep no matter how many times we repeated the assay. And so this was good because this gel allowed us to triage away those samples that always failed. Those samples to the right have the highest degradation profile, so there's just less DNA accessible to the assay. The high-quality data and the low-quality data, we can begin to get some type of a qualitative assessment, meaning if you look at the mean distribution by the little bars or the little arrows next to each lane, you can see that the high-quality data were a little bit bigger in terms of fragment size from the low-quality. And so about 95% of our samples we could triage and understand how they would perform in the assay. We wanted to push it a little bit further and ask the question whether the qubit measurement was really the most accurate, or given these degradation profiles, whether there was an amplifiable fraction, right, the actual amount of DNA accessible to the assay given our amplicon size, if that would be a better measurement. So as illustrated here in the top of this plot, our amplicons are about 180 base pairs. And what we were asking is if DNA is degraded or if it's cross-linked, perhaps our amplicons can't amplify because there's just too much cross-linking or the average size of the fragment is a little bit smaller. So what we decided to do was run a qPCR-based quantification using 3 different amplicon sizes, the first being 41 base pairs, the second being 121 base pairs, and the third being 305 base pairs. And by comparing this to a standard curve, we'd essentially be able to say, what is the concentration of amplifiable DNA at these different sizes, and begin correlating that back to our given amplicon size in our library prep. And so down bottom is data from 7 representative samples where we put in 1 nanogram of DNA based upon a qubit measurement into each of those different qPCR reactions. And you can see to the left at the 41 base pair size, all of the samples show essentially the same quantification. So they're all showing somewhere around that 1 nanogram per microliter. On the y-axis is our concentration in log scale, in picograms, and then on the x-axis is our amplicon sizes. So if we move to the right, you can see at 121 base pair size, the concentration of DNA begins to drop, and by the time we reach 300 base pairs, in some of these samples, the qPCR-based quant is down to as low as a few picograms per microliter. And remember, we put in 1 nanogram per microliter based upon the qubit measurement into each of these assays. So what this is telling us is the DNA is degraded or cross-linked in such a way that it can't amplify at those different sizes. Specifically, at the higher sizes, we see the least amount of DNA accessible to the assay. And so now if we look at this and say, well, at our amplicon size of around 180 base pairs, we actually see that there's about a 10-fold difference in concentration between the best sample and the worst performing sample. And so if we look at that in terms of the quantification, Qubit told us that we had 250 nanograms per microliter going into our next-gen assay, but according to a qPCR-based quant, that would only correspond to about 4.1 nanograms. So very different concentration. And so perhaps this is the problem, and this is where we're starting to see that high background error rate. We're putting in what we think is sufficient amount of DNA, but in reality, it's not enough. So then we took this qPCR assay and said, let's look at the sequencing performance. Here are those 7 samples again, putting in 250 nanograms into the next-gen assay. And what you can see from top to bottom, the bottom samples being more intact and less degraded, that the sequencing read depth, or the column with the red, correlates perfectly to the amount of degraded DNA. The more degraded it is, the less data we're able to generate. And in this column all the way to the right is probably the biggest indicator. These are the number of variants we see in that low allele frequency range. And so the more degraded the sample, the higher the number of false positive variants until we hit that threshold where the DNA is sufficient enough for the assay. So what does this mean? Well, this tells us right off the bat that there's a relationship between the QC at DNA extraction and what we see at the end in terms of the bioinformatic analysis, meaning we need to be able to integrate our QC values from the qPCR quant to really understand what those variants mean that we're detecting at the end of our entire pipeline. And so how do we do that? Well, if this data definitely impacts the final reporting, we need to be able to have systems or tools to be able to do that. And this is where the LIMS come in. And so in our laboratory, we've built sort of a homebrew LIMS system to track all of this. And so you can see all of the different tables and all the different variables that come into the final report. And so this is a huge need in the field, being able to capture all this data. And when we got started, we decided to build our own just because the field wasn't there yet. But as you'll hear in a little bit, there have been advances in terms of the laboratory information management systems that are available that really highlight the importance and equip labs to be able to perform these types of analyses. And so this brings us to one of the last parts of our assay, the bioinformatic analysis. And so I've already shown you some of the relationship between the DNA extraction and how it affects the final variant. Library preparation and different quirks that can happen there can manifest in the same way. And this highlights the importance of trying to find automation solutions to be able to build this pipeline. So this is our custom bioinformatics analysis pipeline that essentially takes the raw reads and generates the final report. And what's highlighted in the yellow are all of the different QC metrics that we pull out as we're processing data that are important to look at when we're looking at the final list of variants and trying to do the final interpretation. And the reason why these QC stats are important is because they tell us about things that happened in the library preparation step of our workflow. For example, one of the first steps of bioinformatics is the process of demultiplexing. As you can see here on the left, we get back a giant file of all of the different sequences that were present on the run. And what we need to do is begin separating those out based upon the different samples and the unique DNA molecular bar tags that are present on each read. We calculate QC at this step because there's residual carryover from run to run that could impact the final variant detection. And so in this case here, you can see that between the previous run and the current run, we actually see about a 1% carryover of residual DNA. So this prompted us to go back and refine our processing for machine washing in between each sequencing run, and is an important QC parameter especially as we want to start working with tumors that have lower percent tumor tissue compared to normal tissue, where the allele frequencies might start bumping up around that 1% to 2% range. If we have just residual carryover or mixing of different samples, that could impact and give false positives. So this is a QC value that we track. In addition, looking at the mapping step, what happens is the reads are taken and they're compared to the human genome. And if a match is found, it's mapped. And if no match is found, it doesn't get mapped. And so we calculate the percentage of reads mapping. And you can see over here in the plot, this was just a representative of 16 different samples, the blue corresponding to the percentage of reads actually finding a match, the red not. And you can see that this is a distribution. And when we went back and looked at our actual processing through the wet lab component, we saw that if there was residual wash buffer right here at the second step of our assay, that directly correlated with the percentage of reads mapping. Essentially, this wash buffer allowed for the oligos to cross-talk with one another and begin creating complex human fusion sequences that would have 20 to 30 base pairs of different fragments of the genome together, but taken collectively would not represent a contiguous stretch of human DNA sequence. After the mapping of reads, one of the next things is what percentage of those reads is actually on target. If you remember back to the schematic of all of the different steps that happen, during library prep is where we're saying, out of the patient's genome, we want to only look at these genes. And so in our case, it's either 33 for our one panel or 47 for the other. And one of the things we like to track is what percentage of data that we've eventually sequenced is actually due to those regions of the genome that we care about. And so again, here's a plot, and you can see the percentage of reads on target varying anywhere from 40 up to 99%. And so we knew that we could achieve these high values, right, the high percentage on target. And so during the piloting and optimization phase, we said, what causes reads to not map on target? And so going back and looking at our workflow, we saw that the temperature at which the DNA was hybridized to the oligos actually impacted that percentage on target. And so these are just a few examples that highlight the importance of capturing the different QC parameters at each of the processing steps, because they can correlate in the final QC report to misperformances and allow you to really fine-tune the workflow to have a highly reproducible assay. And so after defining all of these different performance metrics, we moved into the validation phase, which is where we were able to define our overall sensitivity and specificity of this assay. For us, that meant having a minimum tumor percent of about 10%. So after Mike's talk there, the relationship between the number of tumor nuclei to non-tumor nuclei in a given specimen, 20% is ideal given the current miscalculations based upon the pathologist assessment. But with the advance of these tools, we'll be able to push these limits even lower since we're able to really have a solid estimate of that tumor percentage. And this percentage is really based upon the mutation detection limit. So if we assume copy neutral, where we have 2 copies of every chromosome, if we have a mutation present on one chromosome and 5% of the nuclei corresponding to 10% of the cells, that's where we're able to hit our limit of detection. And so that's for single nucleotide variants, or SNVs, for insertions up to as big as 90 base pairs, and deletions as large as 37 base pairs. The input requirement is based upon the amplifiable quantity, and we can push well below the manufacturer specs of 150 to 250 nanograms down to the 5 nanogram level with the appropriate QC assays. This can all be achieved within a 7 to 10 day turnaround time window. And in terms of reproducibility, up to this point, we've run positive and negative controls in every single run this calendar year, and that's 85 repeats, and we have 100% concordance for all the variants present. And this is really a testament of controlling all of these different parameters from tissue acquisition all the way through the final report generation and incorporating all of that QC into the assay. So what has been the impact over the first 10 months of our clinical testing? Well, to date, we've tested a little over 600 total clinical specimens. As you can see, the lion's share has been in lung cancer, followed by AML, brain tumors, melanoma, and then other tumors. In terms of diagnostic yield, about 75% of them, we're identifying at least one disease-associated change. So this means when we look across the literature that the mutations we see in the specimen have been reported previously as a pathogenic change in some tumor type. And so this is a really high number. Only 14% of cases are coming back normal, meaning we haven't found what it is we're looking for, and this just means we need to expand our content in our future versions of the panel. So in conclusion, I hope that shows you that NGS testing in oncology is definitely very powerful and provides you with a high diagnostic yield, but it's very important to define the relationships between the pre-analytical and the analytical variables as they relate to mutation detection in the bioinformatics pipeline. And capturing all of these different quality-controlled data points is very important in the final result interpretation and really begs the question as to what tools we need to have in place from an automation perspective as well as from a laboratory information management perspective. So with that said, these are the team members that made it happen, and I'm going to turn it over now to Cliff, who's going to talk to you about some of the solutions that are available to accommodate the needs of the laboratory.
All right, Cliff and Andrew, feel free to get started.
Okay. Hi, this is Cliff Hoyt. I run an applications group here in PerkinElmer in Hopkinton, Mass. My group is focusing on how to use our digital pathology tools and other capabilities to facilitate and provide solutions for this exciting emerging field, which needs automation and integration to handle the clinical demands that are expected. So for the next few minutes, I'm going to be talking to you about digital pathology tools, including a tissue microarray robot that can be used for macro dissection. And once I'm done talking about those digital pathology tools, then I'll be handing it off to Andrew Barry, who runs our biomolecular applications group. My group's working very closely with Andrew Barry and his team, which is focusing on the use of PerkinElmer liquid handling automation and other capabilities to automate the process of preparing FFP material for next-gen sequencing. So this slide captures how we're looking at the overall workflow. Samples come in as either FNAs or fine needle aspirates or core biopsies or resections. It's usually the latter. Next, the challenge is to remove from the block or the slide tumor-rich material. And then once that material has been selected, understanding what the percent malignancy of the material is, as Mike discussed earlier. So this workflow starts with scanning an H&E hematoxin eosin view of the slide with one of our color scanners. In this case, we're using scanners from 3D Histech. And then we use image analysis to assess the purity of the tumor and for selecting regions within the block or in the slide to remove FFPE material for processing. If we are removing material from the block, we're using another 3D Histech product. It's a robotic tissue microarrayer called a TMA Master. This machine views the top of the FFP block with a camera and aligns the H&E view of the H&E slide with the top of the block, and then directs the TMA coring drill bit to those areas that were identified by a pathologist as tumor and pulls tissue from the block directly and places that tissue into a PCR tube for subsequent DNA extraction, etc. Andrew will be talking about these steps in slides after mine. So in general, we're at PerkinElmer, we're exploring ways to make this process seamless and appropriate for the kinds of workflows that Mike and Bob need for their work. So we're very interested in hearing from anyone in the audience who might like to learn more about some of the material that we've talked about, and also to help us guide our internal thinking about development directions. So the next slides are about Inform image analysis software. This is the software that Mike Feldman talked about. Here's a screenshot of the interface of Inform. It provides a flexible workspace where you can load in images, essentially any number of images you'd like. These images should be selected to be representative of the kinds of images you'd like to process. And in this case, this is a breast cancer tissue section. It's been labeled with hematoxylin and a progesterone receptor, DAB immunohistochemistry. The next step is to use the computer mouse to draw training regions on the image. In this case, we've decided to segment the image into areas of tumor and stroma. Once we've drawn some training regions that exemplify the tissues, we hit a button in the software called Train Tissue Segmenter. And then in the background, machine learning approaches, including neural networks, are used to find a reliable algorithm that can then be used subsequently to segment other images. And now in this image, you can see how there is now a red and green mask on top of the image. The red is showing where the tumor is, and the green is showing where the stroma is. And then the software has cellular segmentation algorithms that can go into any of the tissue areas and segment individual cells. In the case that we're addressing here, the interest is in understanding how many tumor cells there are as a percentage of all these cells within the image. So in this case, we would use the data that is behind this image. In other words, the numbers of cells in tumor and the numbers of cells in stroma. We would divide the number of tumor cells by the total number of cells in the image to arrive at a percent malignancy. So here we're summarizing the main attributes of this software. It's got a very simple learn-by-example interface. Doesn't require a PhD in image analysis. All you really need to know is be able to recognize the various tissue types. Training of algorithms is quite quick. It takes 10 to 20 seconds to train an algorithm. Another distinctive aspect of this software is that the algorithms we've developed, the features that we use to feed the neural network are quite reliable for clinical variability and heterogeneity. And then once you do have an algorithm, it's remarkably fast. It takes seconds to analyze fields such as this one. So now I'll pass on the mic to Andrew to talk about some of the liquid handling solutions we have for next-gen sequencing.
Thanks, Cliff. So when we think about next-generation sequencing, as a general rule, the faster and less expensive sequencing technologies become, the greater the number of samples that will be processed. And as we heard today, the utility of the data is directly linked to the integrity of the incoming samples, as well as the processes they undergo. PerkinElmer is focused on delivering tools that enable automated, reliable processing of samples from the time the material is harvested until they're loaded onto sequencers. The KeyMagic Perpeto-D is a compact benchtop instrument capable of extracting high-quality nucleic acids from formalin-fixed paraffin-embedded tissues. The Perpeto-D utilizes a series of electromagnetic rods that also rotate. These rods control the resuspension of proprietary polyvinyl alcohol beads that selectively bind nucleic acids. Because suspension is accomplished using the electromagnetic rods and not pipetting, shear forces are minimized, preserving the intactness of extracted nucleic acids. The figure on the bottom right demonstrates typical DNA yields obtained using 10-millimeter slices from a variety of tissue sources. The LabChip GX eliminates the need for manual gels completely to assess DNA degradation. The system uses microfluidic electrophoresis to automate and speed up nucleic acid separations. We recently introduced the genomic DNA assay on the LabChip GX platform, which extends the dynamic range of the system up to 50,000 base pairs, making it amenable for assessment of intact nucleic acids. The Genomic Quality Score, GQS, was developed to create a simple and easy metric to assess DNA degradation. The algorithm behind this metric analyzes the electropherogram from morphology, provides a measure of the intactness of the samples. This provides valuable information regarding the suitability of samples for downstream processing, eliminating costly rework, as well as understanding potential causes for poor data quality. The Janus NGS Express was developed to provide automated sample preparation for the specific needs of a benchtop sequencer. The NGS Express supports all commercial sequencers and reagent kits, providing simple-to-use pre-programmed protocols for multiplexed amplicon panels. These include the Ion Torrent AmpliSeq, Illumina TrueSeq Custom Amplicon, and Agile and HaloPlex. It also supports more complex hybridization-based panels that allow coverage of up to a whole exome using technologies like Agile and SureSelect, Nimble and CCap Easy, and Illumina Nextera Rapid Capture. That having the choice in reagent kits and platforms is important to allow laboratories the ability to tailor the approach for specific gene content, as well as adopt new technologies as they become available. Outside of commercial kits, the NGS Express is an open platform that can be used for standard liquid handling functions with easy-to-use templates for general PCR setup, normalization and pooling of PCR amplicons, primer rearraying, qPCR setup, and many other uses. The inherent value of the automation of these processes is that it provides the necessary throughput, reproducibility, and chain of custody to support NGS workflows. Here's just an example of the reproducibility typical of automated processing on the NGS Express. Figures here represent read-length histograms of amplicon sequences. These were obtained using the ion-torrent PGM. These are actually control HapMap samples assayed against the version 2 AmpliSeq cancer hotspot panel. So the utility of this approach is fairly clear. With this run, we see 100% of the 207 amplicons in the panel covered at 100x. 97% of these are covered at 500x. These amplicons represent genomic hotspot regions that are frequently mutated in human cancer genes. So just to conclude, PerkinElmer strives to develop tools to continually meet the growing demands of translational research, an array of automation and quality control solutions for varying application throughput needs. The goal of PerkinElmer is to provide researchers the tools to develop simplified and streamlined processes to recognize the promise of next-generation sequencing. So with that, I'd like to thank you for your attention today, and I'll hand it back to Tamlin as we transition into the Q&A session.
All right, thanks, guys. You definitely ran through a lot of material very quickly there. Before we start the Q&A session, I want to once again remind everyone about submitting questions. Please feel free to submit questions, and we'll answer as many as we can. I also want to let you know that a post-webinar survey will deploy shortly in the presentation manager. You might need to disable your pop-up blockers in order to view it, but please do take a few minutes and respond. Your comments are really valuable to us. All right, I want to start the Q&A session. I have a bunch of questions for Mike and Bob will get started with you. How does DNA prep affect DNA sequencing results?
That's a great question. The biggest thing is we need to have DNA that's pure and clean, so removed from residual wash buffer, residual ethanol, as well as high enough concentration. And what we've seen is that really the concentration is the critical question for amplicon-based chemistries. If you don't have enough DNA going in, the assay will fail, or we'll see this higher background error rate. So those are the types of things we want to control for.
All right, Bob, also, how variable are DNA prep methods, and do kits vary greatly from one another?
Yeah, so we've tested maybe 5 or 6 different kits out there, and what we've seen is for the column-based kits, we can't control that final concentration as much, so we have to put on a specified amount of rehydration buffer onto the column to be able to rehydrate the DNA. And so we've moved away from those towards kits that are either based on just precipitating the DNA or eluting them off of bead.
All right, Mike, I have a couple of questions for you now. How does variation in tumor composition ultimately affect patient treatment?
Well, I think this is where the linkage between having the anatomic pathologist and the next-gen sequencing lab really makes a huge difference. If we don't know how much tumor we're handing over to Bob in the next-gen lab, it's very possible to come up with a false negative, because the tumor abundance, if you're not quantitating it and it's not linked, that quantitation, to what ultimately comes out of the next-gen lab, you could easily get a false negative. So if Bob wasn't running many thousand-fold reads and I gave him a low percentage tumor, it would simply come up as negative. So a lab that might only be doing 100 or 500-fold, if I gave them a 10% tumor, there's no way they're going to be able to reliably pick up those sequence variations. And so it's the combination of me telling Bob what's in the sample and then him having the next-gen pipeline set up so that he gets the read depth to know that if I have quality DNA, that I can go down to this low in abundance, and here's the abundance that I've been told is there.
All right, Mike, the next question. Will Inform replace the need for a pathologist?
We get that all the time. So is the computer going to get rid of pathologists? And we don't see it as a replacement of the pathologist. We actually see it as a tool the pathologist can use. The systems are simple enough to train. As Cliff showed you, you select an area of stroma, you select an area of tumor, and you hit go. And from that, you can then get a very a reliable, reproducible number of tumor abundance. So these are the types of tools that we can teach a pathologist how to use, as opposed to replacing a pathologist. You still need that trained eye.
I think you reassured a lot of people with the response to that question. Thank you. Bob, we're back to you. Although you did not comment on other library preparation methods such as exome sequencing, do you see similar relationships between DNA quality and overall assay performance?
Yeah, so in our laboratory, we use a variety of different methods. Our clinical assay uses the amplicon-based capture approach, but we do have other test development and research projects where we're initially shearing the DNA, ligating on adapters, amplifying, and then doing the feed-based capture. And we do see a very similar relationship, meaning if the DNA is degraded based upon this qPCR quant, or if the concentration at the size we're shearing is lower, we end up with a higher percentage of duplicates on the back end, which essentially just means that when we went into that PCR step after ligation, there's only a certain fraction of templates that could actually amplify.
All right, Bob, my next question is, does extended extraction and proteinase K improve available DNA from FFPE?
So we do a proteinase K digestion, and we do it overnight at between 60 and 65 degrees because that does increase our overall yield. We've tried doing shorter extraction times or shorter digestion times during the day, and we do get the highest yield overnight. Increasing the concentration of proteinase K can shorten that time if we're really in a pinch and need to get DNA extracted that day because it's a Friday and we have an assay going on Monday and we don't have anyone to work the overnight shift into Saturday. So it definitely does have an impact.
All right, Bob, I have one last question for you. It seems kind of similar to the previous one, but does extended extraction with proteinase K improve amplifiable DNA for NGS?
We haven't seen any correlation between the amplifiable amount, so this would be getting at the integrity of the DNA as it relates to the proteinase K. Really, it's just the overall yield of DNA that's directly correlated to how long we're doing the digest. The variables that affect the quality of the DNA or that amplifiable fraction, we're still trying to figure those out in a few different collaborative projects.
All right, Mike, we have time for one more question, and it's for you. Have you ever tested even longer PK at higher temps like 60 to 65 degrees Celsius?
Years back, not in NextGen, but in targeted amplifications, we did see in 2 and sometimes even 3 days of prolonged PK that we could get more DNA out of paraffin tissue. So I think it's something that might be fun to take a peek at and see on cases where you have really only a little bit of tumor, there's only one sample, you don't want to have to go back and stick the patient again to get more tissue, whether or not something like this might actually be a sort of a last-ditch salvage attempt on some of these types of cases.
Okay, thank you. Our time is up. Thanks for listening in, and thanks for the great questions. This webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to friends and colleagues. Once again, I want to thank our panelists as well as our sponsor, PerkinElmer. Thank you.